My name's Caroline, so I'm joining you from Astrea Bioseparations. I'm in the downstream processing department in the R&D facility in Cambridge, UK. And today I want to talk about some of the chromatography adsorbents that we offer to help with your mAb purification. So the first monoclonal antibody was licensed for biotherapy in 1986. And since then, monoclonal antibodies have grown to become the dominant product class in the biopharmaceutical industry, making up 70% of all the FDA approvals between 2015 and 2021. But monoclonal antibodies are a diverse class of products, particularly now with the advent of humanization of mAbs, the mAb fragments and bispecifics. The products are getting more and more complex, and that means that the purification of these products is getting more complex as well. But that diversity also comes from the different expression systems that you can use for monoclonal antibodies. So as it was said in the introduction, I used to work for BioProcess UK, and we generated IgGs from hybridoma cell lines, but they can also be generated transiently or stably in HEK cell lines, or CHO is very common. And that the expression system will have an impact on the impurity profile of the antibody, and that will affect the downstream processing as well. And also the chemical and physical properties of the antibody can vary depending on the type and subtype of antibody used. So the PI, the hydrophobicity, and the stability, which all impacts the downstream processing. And that diversity means that the purification of mAbs is not a one-size-fits-all process. Instead, we use a toolbox approach. So typically, a primary capture is either protein A for products that have an FC region, or protein L, the products that don't. And following that, there might be 1 to 3 purification, polishing steps, depending on how challenging that product is to purify and what the purity needs are for the downstream, the final application for that product as well. These polishing steps might be a combination of ion exchange, HIC, or mixed mode chromatography in any combinations. I put a few examples on the slide. And they can also be in bind in elute mode or in flow through mode. So I don't have time today to go through every possibility, but I'm going to show some examples of our mixed mode adsorbent used for polishing after a protein A. But just quickly, I would like to show that Astrea offers ion exchange HIC and mixed mode adsorbents in a range of formats. From screening, we use the 0.2 mil robo columns and the 1 in 5 mil pre-pack columns for a lot of our optimization work. And then we can also go through development with our snap columns that are available in a range of diameters and lengths, column lengths, all the way up to pilot and production scale with our evolved process columns. We can also provide columns pre-packed with our adsorbents, the snap and the evolved columns, to help you with your processes as well. So the protein A step that we used for these examples was the Gore protein A membrane. The reason we chose this product was because we had a large volume of CHO IgG from a stably expressing cell line. So we needed to be able to process a lot of material quite quickly. And this allowed us to do that. I just want to touch a bit on the intermediate washes on the protein A. So when you're doing a purification process of your product, you need to get the most out of every step. And one way to do that with the protein A is with intermediate washes, either with high salt, with pH, or additives like arginine. And depending on which intermediate washes you have, that's going to impact your elution impurity profile, and therefore the polishing steps when you're moving on to the next purification in your downstream. So we did a couple of experiments with this protein A Gore membrane with our CHO feedstock, where we looked at the addition of a pH 5 wash prior to the pH 3.4 elution. For this particular CHO IgG, we got a lot of losses of the IgG into that wash step, so this one didn't work for us. But we did find that adding the high salt wash with PBS and 1.8 molar sodium chloride alongside an arginine wash with PBS 0.1 molar arginine, had maintained really good recovery of the IgG, but gave us 3 times less HCPs in the final elution. So that was what we used moving forward. So the material that we generated from that work with the GOR protein A, most of that went on to a viral clearance study that I'm going to go through in the second half of this talk. But we also used some of this for generating examples with our mixed mode and ion exchange adsorbents. So I'm just going to show you now the example with HC Pure. So HC Pure is a mixed mode adsorbent on our Pura bead resin. It has a very unique selectivity due to the unique chemical space that it holds. This diagram here is a principal component plot that plots similarities in the structural groups on ligands, which then infers differences in the chemical space. So you can see that it stands apart from a lot of the other commercially available mixed mode ligands. And that selectivity is also tunable to a wide range of expression systems and target proteins as well. So we've got examples of full-length IgG, IgG fragments for VK and VH, and even AAVs and bispecifics from CHO cell lines, E. coli, SF9s, Pichia, and HEC293 cell lines. But to be able to do that, to be able to have that selectivity over a variable range of expression systems and target proteins, we have to optimize the buffer conditions that we use that mixed mode in. And that's going to be the main takeaway from this talk, is the requirement to do the screening of the purification conditions. HC Pure is also stable to 1 molar sodium hydroxide and can be effectively cleaned and reused for hundreds of cycles over and over again. So it's a great addition to your purification toolbox. Okay, so here we have the broad range robocolumn screen that we used for the CHO HCP, the CHO IgG following the protein A purification. This is a very typical screen that we'll run whenever we have a new product that we want to purify with our HC Pure adsorbent. And this is a broad range looking at the extremes of the conditions that we tend to use on HC Pure. So the pH ranges from 4 to 9, and then we tend to look at between 8 and 18 millisiemens. So HC Pure primarily uses hydrogen bonding and hydrophobic interaction modalities for the binding. So it's not an ion exchanger. You can't predict where the product will bind based on its PI. You have to do the screening to be able to work out the best conditions. So here we used the neutralized protein A elution, and we buffer exchanged that into our different pH and conductivity buffers using a DOE approach, so at the extremes of those conditions. So we have pH 4 with both conductivity at 8 and 18 millisiemens, the same for pH 9, a midpoint at pH 6.5, and we also included the neutralized dilution with no buffer exchange. In this example, we got really good IgG recovery and HCP clearance across the whole range of the conditions that we tested. That doesn't mean you don't have to do the screen. Every time you have to do the screen, this doesn't always happen. But we decided for this product that we would continue to use just the neutralized solution without having to buffer exchange, as it removed the need to do that buffer exchange step. So the next thing we did with that product was to work out how much of the IgG we could load onto our HC Pure adsorbent while it's maintaining below 100 ppm HCPs in the final recovered material. HCPure is used in flow-through mode, so the amount of the IgG that you can process will depend a lot on the impurity profile, how many HCPs there are in your load. So this also needs to be determined every time you have a new product that you're using with HCPure. So in this experiment, we loaded that neutralized CHO IgG with a variety of loadings between 10 and 50 mg of IgG per mil of HCPure. And we saw that even up to 50 mg of IgG being processed on 1 mil of HCPure, we still managed to get way below that 100 ppm threshold. We also did a panel of other impurity tests for this experiment, looking at aggregates, host cell DNA, and protein A leachates. And we had excellent recovery by the end of just a single polishing step. This next example uses a full-length IgG from a HEC 293 cell line. The primary catcher, again, was GOR protein A for this one. In this case, we buffer exchanged the neutralized dilution to pH 6 with 18 millisiemens per centimeter conductivity. So this time, a slightly higher conductivity gave us better results. Again, we ran our IgG on a 1 mil HCPure column. And in this case, we tested for the IgG recovery, the HCPs, and also DNA and protein A leachate. But this time we also had endotoxin measurement as well, because this was produced transiently, so plasmids were used to express the IgG, and endotoxin is therefore more relevant in this case. And we managed again to get really good removal of the HCPs, good recovery of IgG, and the endotoxin was also decreased a lot by this step as well. So the last example that I have before we move on to the viral clearance work is with a VK fragment. So this was produced in E. coli, and as it's a VK, it doesn't have the FC region, so it was the primary catcher was a protein L this time. For this case, again, we managed to load the neutralized dilution directly onto HCPure without the need for a buffer exchange. And you can see that we got great recovery of the VK, reduction of HCP again, and then, of course, we measured the endotoxin in this one as it was produced in E. coli, and we got 99% reduction of the endotoxin as well. So I've shown a few examples now of different expression systems and the different impurities that we can remove with HCPure. So I'm just going to talk now a bit about a viral clearance study that we did in partnership with Cygnus Technologies. So viral clearance studies are important for any antibody that's going to be used as a biotherapeutic due to the clinical consequences of even low levels of viral contaminants. This might be contaminants from the cell lines itself or from adventitious contamination during the purification process. So viral spiking studies are generally used to show the log reduction values across each of the purification steps within a process. And these tend to be with model viruses that will vary depending on what your expression system is. So in the ICH guidelines, they have a whole table of which model virus is most appropriate for your viral clearance study. One of the more common ones is minute virus in mice, MVM. This is a live virus, so it requires specialist skills, specialist laboratories to be able to run these experiments, and also the viral clearance is determined with an infectivity assay. So those viral spiking studies are generally done in combination or with partnerships with specialist CROs, and that can have a high cost associated with it, but also scheduling can be a problem. You can be waiting months in order to be able to get your project set up with a CRO. So that means that viral spiking studies can be left until after most of the development of a downstream process has already been done. And at that point, if you aren't achieving the viral clearance that you need after that viral clearance study, you then have to go back and redesign the purification method, which obviously has a big cost. So Cygnus have developed a BSL-1 compatible mock MVM virus that's available off the shelf and can be used as to predict what the viral clearance will be in those viral clearance studies with actual MVM, as well as an endogenous CHO spiking agent, and the analytical methods to be able to, which are qPCR based, to be able to do those viral clearance studies within your own laboratory, so you have an idea of what to expect when you go and do the viral clearance studies with a CRO. It's not to replace the need to do the CRO viral clearance studies, because they're a regulatory requirement, but it gives you a prediction of what to expect, and also it means that when you're developing your method, you have the viral clearance study in mind from the beginning, rather than it being a tick box exercise once you need that regulatory approval. So we worked with Cygnus to do a viral clearance study with 2 of our adsorbents, our ion exchange Q-Purabid and also HC Pure. So we were interested in this technology because when we're helping our clients, our customers, to use our products in developing their mAb purification processes, we need to advise on the types of experiments that they need to do. So if you're using our HC Pure, then we need to advise how to do the screen. Like I showed before, we always use product recovery and HCPs as our main examples. But we wanted to be able to add in using viral clearance also when you're planning or when you're developing these methods. So this project was kind of a first look-see for us to see if we, like how this technology worked, if we could take this in-house and use it to help our customers. And the way we wanted to do that was provide Cygnus with our adsorbents in our SNAP columns, and then also the feedstock. And then they ran the column runs and the assays. And also they looked at additional things like carryover between runs, and also limit of detection to help us understand how we wanted to use these assays. So what we did was we took the CHO IgG that we purified off that GOR protein A column, and then we did an intermediate purification step with our SP cation exchange. This was in bind and elute mode on an 18 ml column packed into a SNAP column. We loaded at pH 6 and diluted the neutralized dilution to 4 millisiemens per centimeter. And then we eluted off that SP column at pH 6 with 350 millimolar salt. And the reason we did the 350 millimolar salt was because we saw that separated out a lot of the HCPs, which were then later eluted in the 1 molar strip. And then we buffer exchanged that material into 50 millimolar tris, pH 8. And that was the material that we would use as the feedstock for the viral clearance study. The reason we chose 50 molar mitris pH 8 was because that's a pretty standard buffer to use for an ion exchange. And with our HC Pure, as I mentioned, we can't predict what will bind based on PI. We have to run a screen, but for this project, it wasn't in scope to be able to do a screen as we were getting Cygnus to do the column runs. So we wanted to choose one condition that we knew should work well for the ion exchange, and then we just used the same condition for the HC Pure. So to actually perform the viral clearance runs, Cygnus spiked in the spiking agents, either the mock MVM or the RVLP, and then ran triplicate column runs on each of our adsorbents. Both were run in flow-through mode, so they collected the UV peak and analyzed that peak for IgG recovery and also for the viral clearance as well. So you can see just as an example of the chromatogram, most of the product is in the flow-through, and then we stripped the columns with 1 molar salt in either pH 8 for the Q or pH 3 for the HC Pure. We stripped the HC Pure with high salt and low pH because of the multiple binding modes that it has. And then we regenerated the columns with 0.5 molar sodium chloride between each run. So they ran the carryover runs as well, and there wasn't any carryover between each of those runs, so that was good for us to establish that we can use that, these methods later on. So the results of the viral clearance assay then, so on the Q Pura bead, kind of as we would expect for this buffer condition, we got clearance to below the limit of detection for both the RVLP and the mock MVM virus. So the level of the spike kind of determined what the viral clearance, the log reduction value was, because we went to below the limit of detection. Here it was, I have to check my slide, 3.4 log for the 3.5 log for the RVLP on Q and 4.2 log for the MVM. I think we could increase that, so part of this work is, working out how we can use these assays in our lab. So one of the takeaways from this was that we needed to increase the spike so we can up the log reduction values. But the IgG recovery across all of the experiments was really good as well, so around 95, 97%. For the HC Pure in these conditions, we did also get 3.4 log of the reduction of the RVLP. And in this buffer, we didn't see reduction of the mock MVM. And this goes back to what I was saying previously, is that we need to screen HC Pure to be able to work out the optimum conditions. And in this case, we just ran the same condition that we used for the Q. When we did, that's the robocolumn screen that I showed previously, we saw that the HCP removal and IgG recovery was really good all the way from pH 4 to pH 9 and all the conductivities that we tested. So there's a lot of scope there for finding the optimum condition for the MVM removal, which is the next steps that we're going to take in our own labs when we adopt this assay kit in our own labs as well. So this is a bit of a work in progress. So we can improve the MVM reduction either through lower pH, which can drive the hydrogen bonding interaction, or through increasing the conductivity, and that will help drive the hydrophobic interaction. I think that's everything. Okay. So just the last slide I've got here is an overview of all of the products in our mAb purification toolbox. So briefly explain most of these, but we've got the Q and the S Pura bead, strong cation and anion exchanges. we actually also have weak anion exchanges as well, but typically the strong anion exchanges will work. We have the phenol pure bead as our HIC, the main one that we use for mAb purification, but we also have 3 other HIC products, and we provide a selection box of the 4 different HIC adsorbents if you want to screen those. HC Pure, as we discussed, is a mixed mode. We also have a Toxiclear, which is an affinity resin for removal of endotoxin. And then we have Fabsorbent, which binds both the kappa and the lambda light chains on antibodies, and then the columns that we've briefly gone through from the screening to the process size columns. We do have a poster, which is poster location 120, which goes through the viral clearance work. That's where I'll be after this talk, because we've got the cheese and wine session. And then we also have a booth at 515 if you want to talk to any of my colleagues about any of these products. We have one of the Evolve columns there if you want to see one of those as well. So I think that's about a few minutes left for any immediate questions. Thank you. We've got a couple back here.
Thank you. It was such a very great presentation. I had a question on the surrogates you use as a substitute for MVM and XMLV. Has there been a direct comparison with the actual live viruses? And
yes.
Are they comparable?
Yeah, so the question was, has there been any work to show the comparability between the mock MVM and live MVM?
Correct.
Yeah, so Cygnus do have that they have a paper that shows comparability, and then they have examples of the predicted log reduction values from their kits versus actual live MVM as well. So yeah.
Great. Thank you.
Thank you.
Great talk, Caroline.
Thank you.
Just with your HC Pure product,
Yeah.
if I've got this right, so HCP and viral selectivity are tunable, right?
Tunable, yeah.
So that means that you can operate across a wide range of expression systems when the conditions are optimized.
Yes.
That's the take home, right?
That's the take home, yeah. So we get a lot of questions about, what conditions should we run our HC Pure for our target? But unfortunately, we can't answer that. We need you to do a screen for every target, but that's, the downside, but also the benefit, because it means that it is tunable, and it means that we can work across lots of expression systems and lots of targets.
Maybe along those same lines, are there other kind of biochemical surface properties of the proteins that if you could be fully knowledgeable about, if you combined them together, you could directionally make recommendations, or?
Yeah, so I think it's a complicated mechanism, and I'd love to understand more about it. Currently, we do have to do the screens. Like I said, there are some general properties that you can take from that, so like using high conductivity to drive the hydrophobicity, but you would have to still screen it because we don't know how that would impact the product recovery. But yeah, it'd be great to be able to just type in the condition and then know what the result would be.